This next section is on the brain initiative. And really, I've tried to highlight a few stories featuring this new project. So, as all of you know, the Human Genome Project began in the 90s, the very early 90s. And at the beginning of that project, it was not possible to sequence the human genome because the technology was not there. And during the Human Genome Project, sequencing technology was refined and developed, went through a number of modifications, and that ultimately allowed the final completion of that project. We're in a very similar process or stage right now for understanding the brain. The brain is perhaps the most complex machine in the universe, and we don't have the tools to really understand it. And so the Brain Initiative really has as one of its goals to enable the understanding of the brain and to foster technologies that are going to revolutionize our ability to study. And so our four speakers are Ed Lein from the Allen Brain Institute, Lauren Frank from UCSF, Mark Schnitzer from Stanford University, and Andreas Tolias from Baylor. And so I'd like to introduce Ed Lein first. Thanks, Ed.
Is there a pointer?
Yes, and that's and there's a clock down there.
Great. Well, thank you very much for the introduction, Joe, and thank you for the opportunity to speak here today. I'm going to give sort of a whirlwind tour of some of our efforts within the Brain Initiative. How do I advance? How do I advance?
The green.
There we go. Okay. So the gist of what we're trying to do in our part of this Brain Initiative effort is to translate a lot of the advances that have been made on understanding the fine cellular architecture of the mouse brain or other model organisms to the human brain. In the mouse, we have a really emerging understanding of the circuit elements that make up complex circuits in the brain. We speak of very fine types of cells and all of their different phenotypes. In the human, we still largely talk about brain regions and pathological proteins. It's really a very different sort of level of resolution that's possible. And so while the Brain Initiative as a whole is trying to catalog all of the cell types and understand their properties, what we're trying to contribute to this is to move those technologies to their application to human brain. I wanted to sort of mention that there's a sort of sociological element to this as well, that these projects are very sort of ambitious, they're difficult to do, and so they basically require consortia. So these big projects like the NIH Brain Initiative Cell Census Network are actually sort of a series of nested consortia. This is one consortium within a sort of meta-consortium to try to tackle these bigger problems. And it's, in this particular case, trying to bring together experts who specialize in looking at human brain function and human elements of understanding circuits. So the essence of what we're trying to do is to reduce the complexity of the nervous system to a discrete number of cell types that make up neuronal circuits. You could think of this sort of very oversimplified, but as a periodic table of cell types. Can we take this 100 billion neurons in the human brain and reduce it to some understandable and targetable number of entities? A big advance in the field that's allowed this to sort of move forward is the movement into using the genome as the way to characterize cell types and catalog them. And the techniques for doing this that have really revolutionized the field are single-cell transcriptomics. And so whereas the field in general has largely been historically based on categorizing cell types on the basis of anatomy, for example, you have chandelier cells and double bouquet cells and pyramidal cells. Instead, one can categorize them and classify them on the basis of the sets of genes that give rise to those properties. And the techniques of genomics have really sort of come to bear now on brain tissues so that you can start to use this complex set of genes that define the cells as the means of classifying them, and then to use that then as the sort of foundation to understand what all the different properties of cells are. And another advantage of using this sort of genetic paradigm is that genes define the cell types, And now you can use genes to generate tools to be able to target those cell types as well. This is really the big advance in the model organism field. We can now start to approach this using viruses to target cell types in human. So the real key to all of this working is the advent of single-cell transcriptomics. So this is a means of being able to measure all of the genes being used by individual cells. And these are scalable technologies that can be used to look at all the genes used across many thousands of cells. And so you can essentially take a tissue, dissect it into cells, measure all their genes, and now you have a big quantitative vector to be able to classify cell types. Applied to the neocortex, which is the most complex structure, the sort of initial sort of approaches to this defined about 50 different types of cells based on the genes that they use. We've sort of pushed this much further now. And it turns out that another advantage of this is you come to sort of a saturating answer. We can get a complete and accurate and hopefully permanent sort of description of cell types. It turns out that in the cortex, there are about 100 discriminable types of molecularly defined cell types. It also turns out that there are some principles that come from this. So for example, within the cortex, there are two main types of neurons. There are excitatory neurons and inhibitory neurons. The inhibitory neurons, which all come from a single developmental source and populate the cortex, are common across the cortex, whereas the excitatory neurons are actually different in different regions of the cortex. So these types of principles can kind of merge quite easily from these types of data. So this is wonderful. It allows us to sort of, in one fell swoop, be able to just apply scale and categorize the cell types of the brain. But a problem with applying this to human brain is that it requires isolating individual cells. And this turns out to be quite challenging in adult brain tissues. In fact, you can't do it. If you take even a piece of surgical tissue, you can't apply these techniques of dissociation and get the neurons to survive. So it's necessary to innovate on this front as well. And the key to this turns out to be that the nucleus contains a lot of genetic material as well. And excuse me, you can isolate nuclei from even frozen post-mortem brain specimens. We've now put a lot of effort into trying to demonstrate that, in fact, we can get similar results when you cluster these types of data from human or from nuclei and from cells. And this slide is just meant to illustrate that in fact, the results from nuclei versus cells are very, very similar to one another when you do carefully controlled studies. What this means is that we can now apply these techniques to human. And our first foray into this is looking in the middle temporal gyrus, the reason for which will become clear quite soon. And we can get a very similar result. We can get a whole series of molecularly defined cell types that are defined by sets of marker genes, but also the covariant expression of many hundreds to even thousands of genes that discriminate these types of cells. And one point I wanted to make in particular from this analysis is sort of what we've been missing this whole time. So on this row here is the frequency of these different cell types in the cortex as a whole. And what I want to point out in particular is that most of the cell types are rare. So this is part of the problem with trying to define this structure, is that we've been doing needle in the haystack experiments in a milieu where all the cell types are rare. So we finally have a method that allows us to have access to this in human tissues. Another advantage of this is that it allows us to have a quantitative way of comparing across species and try to take advantage of the information we have from model organisms and apply it to the human. So we've been developing new analytical methods to try to align these transcriptome data sets across mouse to human. In fact, it turns out that you can do this quite well. There's so much information in the transcriptome that you can align between species. This is 75 million years of evolution, and we can still align these things. Here's mapping the GABAergic cell populations to one another. And the point I want to make is most of the things are roughly on the diagonal. And even some of the cell types, very distinctive cell types, can be matched one-to-one between species. Sort of taking a broader view on this, within a species or within a cortical region, we have this roughly 75 to 100 molecularly distinct cell populations. When we map between species, we actually see that all the variation is really at these very finest divisions. there really aren't any main branches that are different. So we're quite similar across mammals. But then when we get to those fine branches, there are many distinctions. But what this allows us to do is to sort of build a canonical version of this cell type classification that's about half the level of resolution, where then there's some variation between species at the very ends of this. And this is really excellent news, because what it means is that we can start to apply some of the information from the model organism onto the human classification. So without doing the experiment, we can now say that certain of the cell types are likely to be neurogliaform cells or chandelier cells, or to come from a particular developmental origin that we're unable to measure in the human. Now, this also allows us to align cell types and understand the differences. And I don't have time to discuss these in any detail today, but there are also profound differences between us. We're not just big mice. There are different proportions of cells. There are differential gene regulation within specific cell types. There are major phenotypic differences in otherwise conserved cell types that we see. For example, certain kinds of GABAergic cells, certain kinds of astrocytes have very different phenotypes. But by the transcriptomic criteria, they would be conserved cell types. So the thing that we're trying to do now is to understand what these molecular types actually are. So what is the relationship between the transcriptome and the anatomy and the physiology, other things that we would more traditionally classify cell types on? And this requires having access to living specimens to be able to characterize the functional properties of human neurons. So to that end, we've now established, with the help of some of our collaborators, a human slice physiology platform where we have access to neurosurgically resected pieces of human cortex. These are coming from either epilepsy treatment surgeries or deep tumor removal surgeries, where it's necessary to remove some of the tissue in order to get access to the site of pathology. Those regions of cortex can actually be then taken as for studies, and it turns out that these slices are quite healthy. We're able to do all the things that one can do in a model organism. We can do patch clamp physiology to record the electrical properties. We can fill the cells and get the morphologies. For example, we can then start to model those types of data. And one of the sort of remarkable things that's come out of this analysis is that these tissues are extremely viable. So human tissues can be preserved for several days just on the benchtop. They can be preserved for several weeks in culture. This is very unlike a mouse, where you have only some hours to do an experiment in an adult. So something about our tissues are sort of built to last, which gives us the ability then to do different types of studies in these tissues. So the approach that we've now taken is actually something which has been developed initially by Andreas Tolias here, who'll talk later, which is called patch-seq. This is an embellishment of the patch clamp physiology approach, where you're recording from a cell and you're filling a cell with a dye. Now, at the end, you extract the contents of the cell and you sequence the cell. And if you have a transcriptomic-based classification, you can then map it onto one of those types of cells on that classification. And so effectively start to understand what they are and to annotate this classification. And so this is now sort of well underway. This is a consortium effort across 3 different labs around the world, where we're trying to aggregate data across these groups because each of these has to come from an actual human case. And so we're effectively getting a functional annotation of this molecularly based classification in human. And I just wanted to mention that the data sort of rolling in and we aggregate enough data, we're now seeing a very clear correspondence between some of the electrophysiology properties and these transcriptomic cell types. So this genetic definition seems to have weight when it comes to looking at other functional features of these cell types. Now, I wanted to end by running through one particular story on one particular cell type. Every one of these 100 cell types has an interesting story around it. But this one in particular that's quite interesting is the types of excitatory cells that project from the cortex down to the deep subcortical structures. In some certain regions of the cortex, these are called the pyramidal tract cells, although corticofugal is probably a better description of these cells. And one of the things that's interesting about these cell types is that they're very different in their proportion between mouse and human. So the human cortex is expanded dramatically in evolution, over 1,000-fold, but its subcortical targets have not expanded to the same extent. So you have a mismatch between the size of the cortex and the size of its targets. And what seems to have happened is that the solution to this mismatch is to sparsify that population in the cortex. Any part of the cortex now has many fewer of this particular kind of cell. It's almost 20-fold difference. So this is a big difference in the makeup of the cortical circuit between mouse and human. So this is great, but it also means that this cell population is extremely rare now. And so if we would like to study this, it's very difficult to go study without some way of actually targeting that cell population. So I want to use this as an illustration of another advantage of this molecular approach, which is that we can now, having a molecular definition, start to develop tools to specifically target that particular population of cells. This is another advantage of the single-cell approach. Now we can combine this transcriptome, looking at the RNA, with the epigenome, where we look at the chromatin. Regions of chromatin tend to be open in regulatory regions around those genes. And so we can now identify the regions of open chromatin near genes that are very selective for cell populations and use those to then drive an experimental vector to target those cell populations. And so a nice example of this is that we've now been able to actually develop an enhancer that can target that very, very rare type of corticofugal cell type. This is a sort of hot off the press result here that although these cells are about 0.5% of the population just in that one layer, this virus can now target and give selective expression in that particular cell population. So why is this important? These are also tools for gene therapy. So as we start to gain a better understanding of the cellular specificity of disease, we now have the means to be able to target those cell populations very specifically. Here's just a flashy example of this, but in a human, using two-color labeling that you might mistake for being a mouse experiment, we now have the tools to be able to do this in any cell population that we'd like in human, and this can be applied to disease. And so that's actually the thought that I would like to leave this talk on, which is really these tools open up the possibility of a new cellular lens on understanding brain diseases. And we can start to ask, is there a cellular specificity to particular types of diseases? Are there certain types of cells which are selectively vulnerable or selectively proliferate? What are the molecular pathways perturbed in those cells? And we have the tools to be able to then be able to target and manipulate and hopefully eventually treat these types of diseases. So with that, I'll end. I'd like to thank three sort of groups of people. The first is the neurosurgeons. We've built a network of surgeons to work with. This is work that can't be done without a tight collaboration between surgeons and experimentalists. We have very much a team environment at the Allen Institute. What I've described actually involves a whole series of scientists working on different elements of understanding cell types. And I'd like to finally thank our late founder, Paul Allen, as well as the National Institute of Mental Health for funding this work. Thank you.
That's great. Now, we still have time for questions. So we have time for questions. So please come forward to the microphones if you'd like to ask a question.
It was a fabulous lecture. Just, I'm wondering whether you see a role for case-specific stem cells in for human studies where we don't have human tissue, live human tissue from people. How can we use stem cells?
Yeah, I think that's a great question. I mean, part of the challenge with working with human is that it's difficult to get access to an experimental system. The trouble with stem cells in the absence of a foundational understanding of the real adult system is that we don't really know what we're looking at. So I think the way that I view it is that we're trying to understand the terminal state of development in real human tissues that can be used as a foundation then to benchmark more stem cell types of approaches. So there's a lot of potential in using organoids and other types of culture systems to be looking at actual human cells, but they're going through a developmental process which turns out to have the same time course as real human development. So it's very difficult to get beyond the early stages of development at the moment.
Thanks, Ed. have any of your human cortical samples come from individuals with distinct genetic disorders that might be relevant?
Yeah, that's a great question. So almost explicitly, we've tried to avoid things like that. However, a lot of these tissues come from epilepsy surgeries, and we're now actually starting to sequence all of those individuals and try to understand if some of these have mutations in ion channels and things like that that could be directly related. I do think, aside from finding individuals with very highly penetrant mutations, a challenge with these methodologies is that they cost a lot of money, so it's hard to do population studies. But you certainly can do this in a targeted way.
Thanks.
Over here.
One more?
Sorry, over here.
I'm just curious, you mentioned that transcriptomically you saw similar types of cells between human and mouse, even though there were differences when you got down into the details. It's fascinating that you're now doing electrophysiology on those cells. To what extent does the electrophysiology also line up with what you see in mouse, or does it start to diverge?
Yeah, that's a good question. We're just starting to answer that type of question now. We have a few cases where the genes actually predict pretty significant functional differences between mouse and human, and then we're going and actually finding that that's the case as well when we record. So the data seems to be quite predictive, and we can go in that way. Part of the challenge in doing this serious head-to-head matching between the two is when you get down to that very fine level, it becomes difficult to match up. But I think a key element here is that the genes are predictive. And so we have a measure now of all of the ion channels and all of the receptors. And so we can look for similarities and look for differences and do that in a more targeted way.
Okay, one last question, Roger.
In a disease like Alzheimer's disease, would you be able to differentiate causal changes in the transcriptome from result? If you just looked at early or late samples, is it able to be that sensitive and discriminatory?
I'm not sure if we could get to causal. We can certainly get to correlative. I think we can look across the progression of the disease and look for changes in cell populations, changes in gene expression in specific cell populations. To my mind, causal will require additional experiments of some sort.
Okay, great. Thank you very much, Ed.